Hello everyone, in this video I'm going to be talking about genetic engineering. Now it's something that you're introduced to at Key Stage 4, but you do it in more detail in A-Level and IB at Key Stage 5. So this video is aimed primarily at Key Stage 4 GCSE students. I will do other videos which go into a bit more detail about the actual individual stages involved in the process, but this is just a more general overview. Now, genetic engineering has many different definitions, but I like this particular one because it pretty much sums up exactly what happens, or what it is rather. It is the scientific alteration of the structure of genetic material, and by genetic material we're referring of course to DNA, in a living organism involving the production of recombinant DNA. Now this word here, recombinant, basically means almost like recombined. It's combined DNA from two sources. That's what recombinant means. So making recombinant DNA is combining DNA of two sources. What our ultimate aim is to take the genetic material of one organism and incorporate it into that of another. And what we produce is something called a transgenic organism. So we'll look at some of the advantages of genetic engineering, but also some of the negatives, the arguments against this process. Now, ultimately, the process relies on a few basic principles, a few steps. You take a piece of DNA, you isolate the section that you're looking to incorporate into another organism. So you isolate the section of DNA that you want, and you use enzymes to do that. You then take the second sample of DNA that you're using, and in that case it will be a vector, something that's going to carry your genetic material for you. You insert the selected piece of genetic material initially into that second source, and then you put that vector back into a host. So let's think of insulin as our example. So let's say here is a sample of DNA, and in the center we have a gene, a small section of the DNA that codes for insulin. Now insulin is a classic example taught about in school, because insulin for diabetics, especially those with diabetes type 1, is essential because they have the inability rather to produce insulin from the islets of Langerhans, the pancreas, or the patch of cells in the pancreas that would normally produce that hormone designed to lower the blood sugar. So those with diabetes type 1 rely on insulin injections. Now insulin used to come from pigs. So we'd use pig insulin, but it would be difficult to get and to produce in large quantities. So using genetic engineering, we've been able to produce human insulin. So the first thing that you would do, so we're going to use the insulin process as our example, is you take a piece of DNA, select the DNA, you find the portion that you want, and we've said that this purple section here is the genetic information that we want. And we're going to use enzymes to cut that bit out. So we're going to use enzymes to cut that portion of DNA out. Now those enzymes specifically are called restriction enzymes. So we're going to use restriction enzymes to cut out a section of DNA from a larger sample that we're interested in. Now when we cut that out, what we'll end up making is something that looks a bit like this. Shade it in just to make it a bit more visual. So we cut that out, and we end up making what are known as, so these bits that appear to jut out here, they're known as sticky ends. Now the sticky ends will help us to be able to form our recombinant DNA, to join this section of DNA to the DNA of another source or from another organism. And then, once we've got our gene, our section of DNA with our sticky ends, we then take a plasmid. Now, a plasmid is a ring of DNA, a circular loop of DNA found within a bacteria. It looks a little bit like this. And in previous videos, I think I've drawn it a bit like this. I've shown it as a ring of DNA with two particular genes, one for tetracycline antibiotic resistance and one for ampicillin. Ampicillin being the blue one, tetracycline the red one. Now, I'm not going to go into too much detail about the relevance of that red and blue section in this video, but it is important in the videos I'll make for Key Stage 5, because ultimately the way that scientists check that this process has occurred is by looking at, for example, whether that blue or red gene in the plasmid is still active. But for all intents and purposes, we're just going to say that we take the plasmid from the bacteria. So this is our plasmid. It's a ring of DNA. From a bacteria, and using the same restriction enzyme, we're going to cut that plasmid open. So if we cut the plasmid open, it should look a little bit like this. Now, it's not by any means the best drawing. But you can see that we've got our plasmid ring there with a slot where I'm just putting an asterisk here. So then what we do is we take our selected piece of DNA with sticky ends, so we're talking about our insulin gene, and we insert that into that slot. So you can see those sticky ends on the insulin gene, that small section of DNA that we've originally isolated and cut out. Those sticky ends would match sticky ends that are formed on this plasmid. So if I just colour those in, say, yellow, just to make it clear. So what I've just shaded in yellow there are sticky ends on the plasmid, which will align complementarily to the sticky ends of the insulin gene. So we would actually use the same restriction enzyme that we cut the insulin gene out with to cut the plasmid. If we use the same restriction enzyme, we'll get the same sticky ends forming. So we take that insulin gene, as the green arrow shows, we put it into our plasmid, and then we use another enzyme, and this time we use an enzyme called DNA ligase. And DNA ligase seals those sticky ends together, so we get a nice ring of DNA, a new plasmid, if you like, a recombinant plasmid, if you like, made of two different types or two sources of genetic information. And then we can take that plasmid and insert it into our host, our bacteria cells. So we can put that plasmid back into a bacteria and culture that bacteria. And that bacteria will divide by binary fission, will grow and divide exponentially, and we can harvest all that insulin that it goes on to produce. If we take that bacterial plasmid and incorporate our own insulin gene, then it will ultimately use that gene to produce insulin for us. And all we have to do is culture that bacteria and harvest the insulin it produces. And that, in a nutshell, is the process of genetic engineering. You take a sample of DNA, isolate the gene that you want and cut it out using restriction enzymes. That will form sticky ends. You use the same restriction enzyme to cut a bacterial plasmid or ring of DNA. You insert the chosen gene into that ring and seal it with DNA ligase, and then put that back into a bacterial cell which would then be cultured and harvested to produce the required substance. Now, let's think about some pros and cons. I mean, I've talked about insulin as the example, so that's a major advantage of genetic engineering, that we could produce this hormone in a lab setting rather than using pig insulin, which some people would object to, using actual human insulin and producing it in such large quantities. So other benefits, well, let's think about crops. You can make crops disease resistant. You can make them resistant to cold temperatures, for example, so they wouldn't freeze. You can make them resistant to pesticide, so that they would resist damage from pests. You could make them resistant to weed killer or herbicide. Again, thus reducing or preventing damage to crop yield. One major benefit of genetic engineering is when we think about GM food, or genetically modified food production. In the third world, or developing countries rather, rice is the staple diet for many people. And because they're eating large quantities of rice, they're not getting perhaps the required quantities of vitamin A. And without vitamin A, it can lead to blindness and even death. So scientists have used this genetic engineering technology to incorporate the gene that makes vitamin A. It's called the beta-carotene gene from carrots, to incorporate that gene into rice so that rice now produces vitamin A. Now that clearly is a major pro of this kind of technology. And GM food is also big business, but some people argue that the long-term effects of genetically engineered crops and food is still unknown. And this is where the negatives kind of come in, because it is a relatively, in the grand scheme of things, new sort of technology. The long-term effects of genetic modification and altering the structure of genetic material, we're not certain of. So there is the potential to modify food, but the long-term effects on our health is unknown. It is a relatively expensive process, quite as you might guess, but more than that, there are a number of ethical and moral and social dilemmas that are associated with this. Some people view it as playing God and sort of dealing. So we're thinking kind of eugenics slightly, an altering of populations, because through genetic engineering you can select almost desired traits, desired genes, and equally you could pre-select embryos based on the presence of undesirable traits and undesirable genes, those that, for example, give a predisposition to a particular illness or disease. Some people argue that it is immoral to tamper with genes in the first place. And also, it is impossible to predict, more than that, the ecological consequences of releasing genetically engineered organisms into the environment. So I mentioned about weed killer resistance. If a gene could pass from the organism it's placed in to a completely different one, and the question would then arise, what would happen if a gene for weed killer resistance was actually passed from a crop plant to a weed, and that weed itself developed resistance? So there are a few unknowns with this, but that's where this video is going to end. So all I wanted to do is define genetic engineering and outline its basic process and some of the pros and cons. It is much more complex than that, and in separate videos I will go through the individual sections. For example, when I talked about isolating the DNA and cutting it out, it's a lot more involved than that. And when we look, or when the plasmid gets reinserted back into the bacteria, there's a whole host of assaying, they're called techniques, that scientists employ, first of all, to see if the plasmid is taken up, and then secondly, to see if you've actually been able to what's called transform that plasmid and alter it successfully. So I'll talk about those in alternate videos, but hopefully this gives you an overview of the process, the pros and the cons. Okay, hope that helps.